Good evening, everybody. My name is Konstantin Chernov, and I'm a CEO of MedInnovation, a company based in Berlin, Germany. We are a small diagnostic company developing a new diagnostic platform called Albumin Functionality Test. This platform allows to detect a number of severe diseases like cancer, liver dysfunction, sepsis, and even more. But currently our focus is cancer application, but at the same time we are doing our ongoing study on liver dysfunction and sepsis as well. We're just starting our raising approach, so discussions with investors, our financial team now working on a final financial presentation. And I'm here just to raise awareness about our project and to start pitching potential investors. And the full presentation will come shortly. And I would like to introduce the technology itself first of all, because I guess it's very important to understand what we do. We're investigating albumin. Albumin is the main transport protein in the blood system, and there are some main functions of albumin. In our case, we are interested of the second shown function of albumin. It's the binding and transport of hydrophobic substances. To perform this function, albumin, being a very flexible molecule, changes its conformation. And it was found that these allosteric changes are specific to particular disorders, to targeted disorders. For example, in case of cancer, tumor cells producing a lot of metabolites which entering the bloodstream and being bound to albumin changes its conformation. And this conformation it's like a signature of let's call it signature of albumin. And to identify, classify, and systematize such allosteric changes of conformation of albumin, that's exactly our scientific approach and the idea behind our technology. As an instrument, we're using electronic spin resonance or electronic paramagnetic resonance spectroscopy. It's a quite well-known technology from chemistry, from physics. It's like a brother of NMR, nuclear magnetic resonance, but it's instead of nuclear, it's studying unpaired electrons. So it's a very sensitive technology, a very sensitive instrument which allows to detect ultra small structures. We use a so-called spin label method of ESR, so we're adding a special marker, fatty acid. In our case, it's a 16-deoxyl stearic acid. We use this acid. This acid binds to all 7 binding sites of albumin, which are common for fatty acids. And this allows us to, let's say, observe, to scan albumin molecule from all sides, let's say the full signature. So we are obtaining a lot of information about the function of albumin. That's how the regular signal from ESR looks like, and to get the information we developed from every particular signal, we developed our own software, simulation software, with a very high correlation coefficients of 0.999, which is very precise, and it's like a digitalizing this physics spectra. Just for example, I can show you, it's an illustrative picture, how different spectra look like from a healthy patients and a cancer patients. Even visually, they're different. And these differences are significant and systematic. It's just for illustration. It's very important to understand that we don't measure concentration of albumin, because it's common when we are talking about albumin. Doctors or physicists immediately identify like, okay, it's a concentration. No, we detect function of albumin. It's very important. So from every measurement, we obtaining 45 independent parameters of albumin. It's a biophysical parameters of albumin. For current developed applications, we use just 4 out of them. So it's shown here. So other parameters, it's a subject for further investigations, but now we see for example, on liver dysfunctions that we can use more parameters. So because they're highly correlating to some progressions of diseases. So the procedure itself quite simple. It's a conventional blood test. So we're taking blood serum, the proper preparation, very conventional as well, and it can be executed in every lab. So we're taking blood serum or EDTA plasma, adding our spin-labeled fatty acid, put it in the electron spin resonance analyzer, obtaining spectra, simulating the spectra, and getting the final biophysical parameters, the final results to process the calculations. The turnaround time of the whole procedure is about 20 minutes, so it's quite fast. To provide the test, we developed the full set of instruments. It's an analyzer, digital analyzer, it's an automation system for prop preparation, reagent kits, and the software. Besides simulation software, we also developed a database and cloud system, as well as statistical algorithms to provide additional analysis of obtained data. So currently our database consists from few thousands of records, and we believe it's not just additional asset, and it's also additional IP protection. It's from business point of view. So the cancer diagnosis, as I said, that our most advanced and targeting market is cancer application, and cancer diagnostics market is huge, with the high unmet needs, and especially in the sector of early detection. And our test allows to detect any kind of tumor at early stages. I know that it sounds a little bit like Theranos. I'm used to face with such skeptic, like, okay, we already heard about any kind of diseases detection, but actually that's true, and it's proved by a number of studies. And for example, it's a summary on result of one of our study. From 9 types of cancer, it was prospective and retrospective part of this study. It's a POC study, but quite good, and you see the quite significant differentiation between the healthy group, chronic, and cancer, for example. For the moment, we can say that we're a global tumor marker, so we can detect any kind of tumor. Maybe we're quite weak, I must admit, with the skin cancer, because skin cancer not entering the bloodstream. Its sensitivity is about 50% or 40%, so that's out of scope of our investigation. But on other cancer types, it's a prospective part, so from the primary patients. So the average sensitivity is quite high. So, and it's a little bit lower only in case of prostate cancer. But at the same time, we had a study in Lund University in cooperation with University Clinic in Lund, Sweden, where we compared the combination of our test with the PSA and PSA alone. And we found that actually the combination of 2 tests significantly better than PSA alone, for example. So thus I would like to emphasize that we are not supposed to use our test as a standalone test. We are a good partner for conventional diagnostics tests, for example to PSA or others, and we see our niche as a first link in the diagnostic pathway. And the most proper way is a colorectal cancer. And this is our aim of our upcoming study, a validation study, which we're supposed to provide in the US. And here you can see the also differentiation between different stages and also a healthy patient group. It's a summary from 3 studies which we did on colorectal cancer. So the advantages of our test: we are high sensitivity and specificity. We are relatively inexpensive. It's a 50 euro per test. We have fast to use, non-invasive, And so we see that we would be a very good candidate for triage situation, for example, on colorectal cancer. Now we are aiming to go this study. So, in spite of we are a global tumor market, we had some POC studies on differentiation, different types of cancer, and we have found that our test can differentiate. We just having a lack of data on that, and this is exactly another aim of our upcoming study, to prove these primary results from our POC project. So here you can see the result as an example on RSC curve on colon cancer. So out of 300 patients, it was 41 colon cancer patients. And our test can significantly differentiate 40 colon cancer cases out of 300. So I hope that soon we can also discriminate the types of cancers. So this is our go-to-market strategy for now on cancer. So all these studies are established with the group partners in Germany. In Germany, we supposed to complete this validation study till the end of 2023. It's a CRO, so we know exactly clinics. It's a multicentric study. In the US, it's a screening on colorectal cancer. We have a very reliable and strong partner from the top 5 healthcare provider. I can disclose the name under NDA if someone would like to go into details. So we guess it will take a little bit longer because we will need a 510(k) certification. We have a CE mark in Germany, so we are a certified producer of medical equipment, but for the US we will need to certify it in the US. That's why we expect that it might take a little bit longer. And also the study in Israel, as far as one of our member of advisory board, Professor Nadir Aber, he's a chief of Integrated Cancer Prevention Center, and we believe that leading by him in Israel, we could achieve also good results. Just last year, we completed the first study in Israel, and we got a quite promising results on that. So this is a cancer application, and just few words about two more. It's a liver and sepsis, as I mentioned in the beginning. So here is some preliminary results from our liver studies. So differentiation between different groups. And this is a very fresh from the last year, our study in Bologna University, which we did with Professor Caraceni, also a member of our advisory board. And we found that we could significantly improve the prognosis for AD, acute decompensated cirrhosis patients developing ACLF, for example, acute and chronic liver failure, as well as to provide this significant information regarding mortality risk. You see here we can differentiate two groups by a Kaplan-Meier survival curve. So it's on liver. And now we do a study with the Fraunhofer Institute in Germany, Rostock University, Professor Minzner, on sepsis. We just started, and we expect to get the results till the end of this year, I guess even earlier, maybe till the end of summer. So this is some of our preliminary results on sepsis. So that's our clinical pipeline, clinical studies pipeline. We are raising, as it's shown in this slide now, 8 million for our cancer pipeline. But also we could consider some splitting on particular programs, and we would be happy to find potential partners who will support us. And yeah, of course we will provide more information by request. Here's our scientific advisory board for the moment. We expect that some more people will join us shortly due to ongoing study, and as soon as we finalize it. So Professor Nadir Aber, as I mentioned, it's in Israel. It's the chief of Integrated Cancer Prevention Center. Professor Paolo Caraceni, he is a member of steering committee of CLIF, Foundation of Consortium of Liver Failure in Barcelona, but he's based in Bologna University. And Professor Rajiv Jalan, with whom we also do hepatological studies. He based here in UCL, in London, and I'm really happy to work with these guys. They're brilliant scientists, and really we believe that we achieve great results together with them. We have small team, absolutely devoted people who works together, who is working together since many years. It's a well-knit team, and just I'm limited in time, and just I need to finalize. So if you have any questions, I would be happy to answer, and so